That's a very kind introduction. Can everyone see my screen?
Yes, perfect.
Great, thank you. Well, I would love to be there presenting to you in person today, but this is a virtual symposium and I'm presenting to you from my office here in the Princess Margaret Cancer Research Tower at the University of Toronto and Princess Margaret Hospital. And I'd like to speak to you today on the role of gain of chromosome 1q in multiple myeloma and other cancers, as revealed by paired profiling of RNA transcriptomes and DNA copy number variations in single cells. And I'll talk to you a little bit about the methods that we developed to do this work. So if we look at the genomic landscape of human cancer, we see a highly recurrent pattern of chromosomal copy number variations, or CNV, including gain of chromosome 8q and deletion of 17p and gain of chromosome 1q. And although the location of the c-Myc oncogene on chromosome 8q was described by Ricardo Della Favera in 1983, and the great Vogelstein demonstrated the existence of the TP53 gene on 17p, also in the early 1980s. It's really the role of gain of chromosome 1q in human cancer really remains much less well understood. And here we're looking at a range of different common tumor cancers, more than 3.5, nearly 4,000 human cancer genomes. And you can see that chromosome 1q gain almost rivals chromosome 8q gain in terms of frequency across these different cancers. Chromosome 1q has been described in multiple different human cancers, including most of the carcinomas, head and neck, thyroid, lung, esophagus, liver, pancreas, colorectal, bladder, breast, ovarian, uterine, cervical, and prostate carcinoma. It's also been described in the sarcomas, Ewing sarcoma, mesothelioma. It's been described in melanoma, and it's been described in the blastomas, pediatric gliomas, medulloblastomas, neuroblastoma, retinoblastoma, and ependymoma. And it occurs with frequency as well in the hematologic cancers, including non-Hodgkin's lymphoma, large B-cell lymphoma, myeloma, myeloproliferative neoplasms, AML, and ALL. In fact, it's always easier to list the human cancers that don't have chromosome 1q gain than those that do. And whilst not all of these cancers have a high frequency of 1q gain, many of them do. So if we look at non-small cell lung cancer, adeno or squamous, we see that 1q gain occurs in more than 50% of these tumors. If we look at breast cancer, 1q gain is one of the most common copy number variations in breast cancer, paralleling 8q gain or c-Myc. In liver cancer, again, 1q gain is highly recurrent and parallels gain of 8q, the c-Myc locus. And in high-grade pediatric glioma, gain of 1q is one of the most common copy number variations, rivaled only by platelet-derived growth factor alpha. So 1q gain is common across many human cancers, and yet its role is poorly understood. 1q gain is not only common, it's also a poor prognostic marker. If we look at Ewing sarcoma, overall survival of patients with this sarcoma is significantly adversely impacted by presence of 1q gain. In pediatric high-grade glioma, 1q gain is again a negative prognostic marker. In retinoblastoma, we can see up to 70% of tumors that are the worst of the worst, the relapsed extraocular retinoblastomas. These tumors have 1q gain. In non-small cell lung carcinoma, if we look at 1q gain, or this study that looked at CNVs and their association with relapse, found that only 1q gain was associated with relapse in non-small cell lung cancer. In breast cancer, if we look at XO1 as a marker of 1q gains, it also has an adverse impact on survival. And in esophageal cancer, poor prognostic cancer at 1q gain, patients in the small study that have 1q gain will do much worse than those that did not. Even in good risk, or supposedly good risk tumors such as Fable cystology Wilms tumors, where not many children are dying these days of this tumor, thank goodness. The ones that are typically have 1q gain. And if we look at medulloblastoma, patients without 1q gain are doing much better than those with 1q gain. So 1q gain is frequent in cancer, but also a high risk phenomenon. If we look at multiple myeloma, plasma cell tumor, and we look at the copy number variations across more than 1,000 myeloma samples, we see that 1q gain is a highly recurrent feature both in samples driven by translocation or samples driven by hyperdiploidy, trisomies of the odd number chromosomes. You can see that in the translocated group, chromosome 1q gain is by far the most common copy number gain in these tumors. And even in the hyperdiploid myelomas that are driven by these odd number chromosome trisomies, 1q gain is the most common secondary copy number gain. If we look at the biology of myeloma and its transition from a pre-malignant monoclonal gammopathy of undetermined significance through to smoldering myeloma, through to symptomatic active invasive myeloma, Leif Bergsagel and others have described that 1q gain is the most common copy number gain that occurs during this transition from NGUS to myeloma, suggesting that it may have well-planned disease biology. And John Shaughnessy and others back in 2006 already described that amplification 1q21 was virtually absent in NGUS, but it was present in nearly 43% of patients at diagnosis and continued to be acquired during disease course, so that at relapse, up to 70% of patients had 1q gain. So this is an abnormality that increases in frequency during disease progression. Hervé Yvette-Louiseau of the IFM demonstrated that 1q gain studied as a single abnormality is associated with a significant 4-year loss in overall survival for myeloma patients. And a number of studies have demonstrated that irrespective of how you treat myeloma patients, either with autologous stem cell transplant or with novel agents, recombinant proteasome inhibitors, or with both, or with monoclonal antibodies, the outcome is always worse in patients who have gain of chromosome 1q. So what is 1q doing in cancer? What is it doing? It's there, it's frequent, it's a poor prognostic marker. There's been a number of studies that have tried to address this. And some studies have tried to address this by defining the minimal region of gain, or the shortest region of gain associated with 1q. However, there's a problem with this approach in that most tumors have full arm gain of chromosome 1q. There's very few tumors that have only small regions of 1q gain, and it's not clear that these overlaps necessarily represent a shortest region of gain. So this is hepatocellular carcinoma, liver cancer. This is 6 different cohorts of breast cancer, where we see that 1q gain is gained as a full chromosome arm. And some of the pediatric tumors, perhaps the situation is not quite as clear. Retinoblastoma, most of the cases are full arm chromosome gain, but perhaps in pediatric hyperdiploid ALL, there's more of a tendency to centromeric rather than telomeric gain. Ron Pino and others have looked at have you attempted to use shRNA to look at functional studies of 1q21, so just one cytoband segment on chromosome 1. And in doing so, they knocked down one gene at a time to identify genes that were important on 1q21. But it's important to note that this approach really identifies essential genes rather than genes that may, through a single copy number gain, promote oncogenesis. And indeed, most tumors, as I noted, have full chromosome arm gains rather than just 1q21 gains. So it is possible to look at the transcriptional consequences of gain of chromosome 1q by taking a tumor cohort. In this case, this is the University of Arkansas Medical School Total Therapy 3 cohort of 350 myeloma tumors. It's possible to take those tumors and divide them into tumors that either have gain of chromosome 1q or don't. And of course, when we do that, we can readily identify tumors that have gain of chromosome 1q. But we quickly find that there's a problem with this approach, and that there are a number of other non-random co-segregating chromosomal abnormalities that go along with 1q. So in this population of tumors, we see not only enrichment of gain of 1q, which is what we're selecting for, but we also see that these tumors have more AQ gain. They have more deletion of chromosome 1p and more deletion of 13q. And there's a bias against the hyperdiploid myelomas with the trisomies of odd number chromosomes from developing 1q. So these have less gain of chromosome 1q. There's also a bias in the myeloma subtypes that develop gain of chromosome 1q. So myelomas that have translocation 414, which dysregulates MMZ or WHSC1, are more enriched in the cohort that have gain of 1q, whereas the hyperdiploid myelomas and the myelomas with Tiedemann 14 translocation, which dysregulates cyclin D1, have less chromosome 1q gain. So when we come to look at these two populations by gene set enrichment, yes, we can pull out all of the 1q cytoband gene sets nicely in this population, but you can see here the striking divergence in myeloma subtypes that goes along hand in hand with that segregation of tumors by gain of chromosome 1q. And here you see that MYC target genes are also enriched in the tumors that have gain of chromosome 1q. And as a result of this, there are significant transcriptional consequences from MYC enrichment and from these divergence of myeloma subtypes between these two cohorts. And so when you try and look at the functional gene sets that differ between these two populations, you're no longer able to determine which of these gene sets is driven by the presence of gain of chromosome 1q and which is driven by the trisomies that are present in this subgroup or the overexpression of MYC in this subgroup. So we're at a loss to determine what the effect of 1q is on this tumor population. So we attempted to address this problem using single-cell RNA sequencing. We actually use the 10x Chromium system. There are several systems out there. And basically, single-cell RNA sequencing is a way of counting the number of RNAs produced per gene per cell. That's it. Before discussing our results from RNA sequencing, I'd just like to take you through the methods a little bit. So the first step in any single-cell RNA sequencing analysis is really to normalize the data. This is how the data comes out off the machine. Once you've got your matrix, each of these columns is a cell. Each of these columns is a single cell, and each of these rows is a transcriptional level for the genes that are being expressed in that cell. So this might be 1 transcript, 2 transcripts, 3 transcripts, 4 transcripts, all the way up to hundreds or thousands of transcripts. This is a log scale. And these are large cells, and these are B cells, and these are all cells essentially from the same lineage, B cell lineage, although these are transformed larger B cells. So there's a problem when we come to analyze this data. You can see that this one gene is not being expressed at the same level. There's really a difference in the depth of transcriptome profiling that we're able to achieve in these small cells versus these large cells over here. So it needs to be normalized. You can normalize to the library size, the transcriptome size of each cell, and this is called transcript per million TPM approach. And this is actually the approach that was used early on for single-cell RNA sequencing in a lot of studies. And this looks pretty good, right, in terms of normalizing that data to this data. But the problem comes when you try and look at the midpoint of, for example, the set of housekeeping genes. And if you just take the average of all the housekeeping genes using a broad list of housekeeping genes, and just look at that midpoint of that list, you can see that the midpoint for the different cells is highly divergent. While you expect that any single housekeeping gene might go up or down according to which cell you're looking at, or even B cells versus plasma cells, when you start talking about 100 genes and looking at the midpoint of those 100 genes, that midpoint should be much more uniformly aligned across these cells, which will be lineage-derived cells. So you can see there's a problem here with some of these cells having very low midpoints and other cells having much higher midpoints with this normalization. So there are other normalization strategies out there. There's SCRAN. SCRAN is doing a bit better here. You can see in terms of pulling the midpoint of gene expression up on these cells. This is a little bit tighter. SCRAN works by developing combinatorial pools of cells, and then develops a system of linear equations to align all of these pools of cells to one another. Other people have taken the approach of using SCONE, which is a quantile method. And you can see here, the midpoint of gene expression across each of these cells is looking a bit tighter again. SCONE works by basically taking, ranking the gene expression in each cell, and then taking that gene and assigning a new expression value based on a reference distribution. So you lose the original distribution of expression within the cell, but all of the ranking of the genes is the same, and now each cell fits a reference distribution. You can see that the effect of this is really that highly expressed genes, which are some of the most interesting genes in the cells and most accurately expressed by single-cell RNA sequencing, are basically reduced to a single line as they fit the reference distribution. So we developed RTAM1 and RTAM2. This is Rodger Tiedemann Ali Madikpour 1 and 2 methods of normalization. And basically, these work by taking the highest expressed genes in each cell, which are the most reliably expressed genes with the tightest resolution in terms of their expression. They're expressed often at hundreds or thousands of transcripts. And using those genes to align the distribution curves for each of the cells, and we developed an exponential, a logarithmically sensitive adjustment factor to bring up the adjustment for each cell to a standard curve. And you can see that using that method, we achieve, which is based on the highly expressed genes, we achieve much better alignment of the cellular transcriptomes, even at the midpoint of gene expression. This is RTAM2, which is a similar approach in terms of using the top expressed genes for our normalization, but basically instead of using ranked genes to make the corrections, we actually use gene intensity. So each gene intensity is not changed, but scaled proportionately to achieve the normalization here. And comparing these different approaches, for these two different samples, you can see that if we look at just the midpoint of housekeeping genes, the RTAM1 and 2 are performing better. Even if we look at the midpoint of all genes that are commonly expressed across all of these cells, so there's all genes, the midpoint is much tighter with RTAM1 and RTAM2. So we believe this is an improvement in single cell normalization approaches that can pay dividends when it comes to analyzing single cell RNA sequencing. So we also developed sciCNV. sciCNV is a method for inferring DNA copy number variations in single cells from RNA sequencing. So this is a single cell RNA sequencing profile where the RNA in this particular cell has been normalized to normal cells of the same lineage. We can take that RNA profile of a single cell and we can convert it into an estimated DNA copy number profile for that single cell. So this is the underlying DNA copy number that we estimate from RNA expression. And if we compare that with the ground truth copy number variation profile of the tumor derived from DNA sequencing of pooled bulk tumor cells, we can see that there's a high correlation between our single cell RNA sequencing method and the ground truth tumor CNV profile. We can do this across hundreds of different cells, giving each one of them a single cell CNV profile from single cell RNA sequencing. And when we do this across multiple samples, we can see that we can pull out virtually all of the copy number gains and losses that are present in each of these tumors as defined using ground truth DNA sequencing. And we can do this in a way with much more accuracy than prior methods that have been developed to do this, with greater sensitivity and specificity and receiver operating characteristics than prior methods. So now what we can do is we can take single cell and we can do single cell RNA sequencing, and we can use our sciCNV method to look at the DNA copy number variation profile of that single cell. And from that single cell, we can also look at the RNA transcriptional profile of that single cell. And if we do this across a range of different cells, we can start to match up the effects of a copy number variation on the transcriptional profile of cells. So to look at the role of chromosome 1q gain in multiple myeloma, we screened 30 samples using sciCNV. We performed some RNA sequencing and performed sciCNV. And here are 10 samples that all have gain of chromosome 1q in the tumor sample. And I want to focus on these two samples here because these are important. These are two samples that have subclonal gain of chromosome 1q within the tumor. Essentially, these two samples give us models of primary tumor cells that are isogenic with their sibling cells. They have exactly the same copy number profiles with sibling tumor cells, but which diverge at a single copy number variation, gain of chromosome 1q. So we can take those two samples and we can look at the differential gene expression of cells that either have gain of chromosome 1q or have wild type 1q. And we can take that differential gene expression profile, those differential gene expression profiles, and we can do gene set enrichment analysis to work out which gene sets are being enriched in the cells with gain of chromosome 1q. This is a control analysis. We started with an analysis of chromosome position cytoband gene sets. And you can see that when we compare these cells with these cells, or these cells with these cells, that in the cells with gain of chromosome 1q by our sciCNV approach, we pull out all of the chromosome 1q cytoband gene sets by gene set enrichment analysis, confirming that we can do gene set enrichment analysis on these populations and pull out accurate results. And you can see the gene set enrichment analysis result here. So what about functional gene sets? Well, we made the striking finding that amongst cells with gain of chromosome 1q, we could see enrichment of mitochondrial respiration or oxidative phosphorylation in these cells with chromosome 1q gain. And this occurred in both samples analyzed independently. And we think this is a genuine program that's been enriched of mitochondrial respiration because the gene set that's next most enriched is a gene set that deals with reactive oxygen species, which is a byproduct of oxidative phosphorylation. Interestingly, in the cells with gain of chromosome 1q, we also saw suppression of type 1 interferons, interferon alpha and gamma, in both samples analyzed independently, and suppression of immune rejection signatures within tumor cells. We now move to a much larger set of genes, the curated gene set list of more than 3,500 gene sets offered by the Broad. And if we do gene set enrichment analysis, again comparing these cells with these cells or these cells with these cells, we can see that the cells with gain of chromosome 1q again show marked enrichment of oxidative phosphorylation gene sets. In fact, of these more than 3,500 gene sets, 8 of the top 15 hits from both of these two tumor samples analyzed independently were identical, revealing that we have really good precision with this approach in terms of identifying gene sets that are enriched by gain of chromosome 1q. So we saw oxidative phosphorylation gene sets, electron transport gene sets, as well as a breast cancer 1q21 amplification gene set. In terms of gene sets that were being suppressed in the 1q gain cells, again, we saw a number of different interferon signatures and inflammation signatures that were being suppressed in the cells with 1q gain. See that here. So oxidative phosphorylation genes are amongst the genes that are most upregulated in the cells with 1q gain. You might think that that's because those genes are perhaps located on chromosome 1q, but that's not the case. If we look at the leading edge of genes that are of oxidative phosphorylation genes that are upregulated in the 1q gain cells, you see that most of these genes actually are located on other chromosomes, not chromosome 1q, pointing to the fact that this is the whole program that's been upregulated and not just a partial program located on 1q. If we look at some functional data, some seahorse assay data, and this is not even our data, this is just reanalyzed data from an Emory group reported in Nature Communications recently. We reanalyzed their data and segregated their cell line data according to the presence of gain of chromosome 1q in the cell lines or wild type 1q. You can see that oxygen consumption significantly increased in the cells with gain of chromosome 1q, both at a basal level and maximal level and in the spare respiratory capacity, confirming that these cells are performing more oxidative phosphorylation. We look at genes that are suppressed in cells that have gain of chromosome 1q. We make the striking observation that TDIT4 or RED1, which is a cellular stress response gene, a gene that typically might go up in response to energy stress, for example, goes down in the cells with gain of chromosome 1q2 gain of chromosome 1q. So what we think is happening is that these cells are performing more oxidative phosphorylation, producing more ATP, and their energy stress is going down. And because their energy stress is alleviated, DDIT4 is going down in these cells. We can map the chromosome 1q genes onto the gene sets that are being dysregulated in cells with gain of chromosome 1q. This helps us define which of the genes may be driving these phenomena. So, for example, we can see that the interferon gene sets are probably going down because of upregulation of ADAR from chromosome 1q21. But we also see that the immune response genes are likely going down as a result of upregulation of complement regulatory genes from several different chromosome cytobands on 1q. And that oxidative phosphorylation has been upregulated in 1q cells as a result both of MCL1 upregulation, but also as a result of upregulation of multiple oxidative phosphorylation genes that are dispersed across the chromosome 1q. We believe that it's this contribution of all these different genes that are dispersed across multiple cytobands of chromosome 1q that accounts for the fact that many tumors show gain of the whole arm of chromosome 1q rather than simple focal amplifications. So MCL1 is well known for its anti-apoptotic function, but actually MCL1 also localizes to the mitochondrial matrix and upregulates oxidative phosphorylation. And it does this in cooperation with c-MYC. And I'll come back to that in a minute. We can look at the programs in 1q cells that correlate at a single cell level. So if we look at single cells and the level of oxidative phosphorylation in those cells as compared to their 1q signature, you can see that OXPHOS is going up with 1q, ROS is going up with 1q. Glycolysis is a bit variable. It's either staying stable or going down as oxidative phosphorylation is going up. And these interferon signatures are going down at 1q. If we come back now to looking at tumor cohorts, so cohorts of large numbers of, for example, myeloma tumors, and come back to this problem with the University of Arkansas Total Therapy 3 data set. If we do that gene set enrichment analysis on these two different tumor cohorts with and without 1q, we can see at the top end of this gene set enrichment analysis, gene sets in the 1q tumors such as proliferation markers and MYC. We don't think that those gene sets are specifically being driven by 1q. We think that those are driven by, for example, the co-segregated AQ24 abnormality. But a little further down on the gene set enrichment list, we do see oxidative phosphorylation and ROS. We think those are the abnormalities that are specifically being driven by gain of chromosome 1q. And across these 350 tumors, we also see the suppression of interferon response and inflammatory responses in these bulk tumor samples that have gain of chromosome 1q. If we try and look at the effect of chromosome 1q on survival, we can do this using the COMPASS data set. This is a data set of more than 1,100 myeloma patients and tumor samples. You can see, we can recapitulate the IFM data and show that gain of chromosome 1q is associated with significant loss in overall survival, again, 3 or 4 years in this US-based data set. If we now forget about 1q for a minute, but just look at the effects of the cellular programs that we think are being dysregulated by 1q, if we just look at oxidative phosphorylation in these tumors, we can see that if the tumors have more than average oxidative phosphorylation, those patients do much worse. And so we think that oxidative phosphorylation, which has been upregulated as a result of 1q, is likely the main mediator of the adverse prognosis of 1q in multiple myeloma. Now you can see how similar these two curves are. Having said that, even if we look at suppression of interferon responses, this is weakly associated with adverse survival as a suppression of inflammatory responses in these tumors. So these it's likely that all of these effects are contributing to the adverse prognosis of patients with gain of chromosome 1q. If we look at overall survival, we break this down according to 1q or the presence of dysregulated MYC. We see that 1q alone is not that bad. Does not have such a bad effect on overall survival. Whereas gain of MYC has a significant effect on overall survival. But really, if you add gain of chromosome 1q to gain of MYC, now is where you really start to see patients with adverse prognosis and poor survival. What we think is happening here is that the gain of 1q is really cooperating with MYC to upregulate oxidative phosphorylation. So if you look at the corresponding tumor samples from these patients, you can see that there's this stepwise increase in oxidative phosphorylation that's occurring as tumors gain chromosome 1q, or gain MYC, or gain both. Well, what about other cancers? We actually analyzed a series of pancreatic adenocarcinoma samples and have looked at their sciCNV profiles, and we're able to identify a sample here that has subclonal gain of chromosome 1q. So you can see subclone 1, no 1q gain, subclone 2, so it shows a 1q gain. Subclone 3 does some other stuff, and then subclone 4 actually appears to gain an extra copy of chromosome 1q. So if we analyze those different subclones, and we will compare them to the subclone 1, which is our baseline control sample, all of these other subclones show this, 2, 3, and 4 all show this enrichment in 1q gene sets, as expected. And if we look at our sciCNV estimated copy number of 1q copy number, we see that in subclone 1, we're diploid, we've got 2 copies of 1q, and 2 and 3, we've got 3 copies of 1q, and in subclone 4, we've got 4 copies of 1q. So if we now do some more gene set enrichment analysis, we see that with subclone 1 here on the right and subclone 2, 3, or 4 on the left, there's this stepwise progression, stepwise increase in the amount of oxidative phosphorylation that's occurring as these subclones evolve from 1 through to 4. So subclone 2 with gain of chromosome 1 has an increase in oxidative phosphorylation, but by the time you gain an extra copy of chromosome 1q, your oxidative phosphorylation is markedly enriched in those cells. Similarly with interferon suppression, in the original subclone 1, in subclone 2 with 1 copy number gain, there's a decrease in interferon signatures. But by the time you get through to subclone 4 with 2 extra copies of chromosome 1q, we see this very strong suppression of interferon signatures. So the actual copy number of 1q is having an impact here in pancreatic adenocarcinoma with stepwise increases in oxidative phosphorylation, stepwise decreases in interferon. We can actually take the sample and can look at the difference at the subclonal evolution, the CNVs that are occurring at each step, and see the pathway changes that are occurring within each of these subclones. Well, if we take a step back and then look at the TCGA data and come back to our cohort of nearly 4,000 tumor samples with gain of chromosome 1q, and then we segregate those samples out according to whether or not they have chromosome 1q gain or not. And we do gene set enrichment analysis on those tumors according to the different tumor types. We can see that in many tumor types, we see this oxidative phosphorylation being upregulated, often to a large extent or sometimes to a smaller extent across all these different tumor types. And in other tumors, we see the interferon gamma and alpha signatures being suppressed virtually across the board in all of these different tumor types with gain of chromosome 1q. So this is not just a myeloma phenomenon, this is a pan-cancer phenomenon. And I think goes a long way to explaining what's going on with chromosome 1q gain in human cancer. So this is our model for what chromosome 1q gain is doing in human cancer. Of course, there are, MCL1 is having potentially some some anti-apoptotic effect. You have to remember that what we're talking about here is single copy number gain of all of these genes. So only a 50% increase potentially in the expression of the genes on this chromosome. And a more important effect appears to be upregulation of oxidative phosphorylation or mitochondrial respiration, driven in part by MCL1 likely get into the matrix of the mitochondria, and also by a number of the oxidative phosphorylation genes contributing to this increased pathway. And because oxidative phosphorylation yields 32 ATP for each glucose that goes down that pathway, versus only 2 ATP for glycolysis. You can see that just a little bit of increase in oxidative phosphorylation, as might occur with a 50% increase in expression of these genes, might go a long way to increasing ATP supply to the cell and alleviating energy, stress, and reducing DDIT4. So we think all of this is contributing to increased tumor metabolic efficiency, and which is one of the benefits of chromosome 1q to human cancer. But also we're seeing that ADAR is going up and interferon responses are going down, and complement regulatory genes are going up, and a number of inflammatory response genes are going down. And that, as an extra added bonus, 1q is also reducing tumor immune rejection and inflammation, and that's helping. It's helping immune evasion, and all of this together is enabling these tumors to develop, to progress from being pre-malignant into being malignant, and contribute to the adverse prognosis of tumors that acquire this abnormality. And so I'm a little early, but with that, I'd like to wrap up and thank all the people in my lab who contributed to this effort, and particularly Ali Madakor, who did a lot of the analysis with me. Our other contributors to this work, our collaborators on other projects, my mentors, including Keith Stewart, Leif Briggsagel, Aaron Schimmer, and John Dick, and of course the patients and our funders. Thank you very much for inviting me for the talk. Thank you.
Thank you so much. It's very interesting talk. Do we have any question from the audience? Please raise your hand or type your question in the Q&A. I might just ask one general question to give people more time to think about their question. So one question is that I think for the single cell RNA-seq analysis, the normalization method you actually developed in your paper, have you compared with the Surat default function like SC transform, and then this is basically question one. And the other question is that so in terms of like a single cell RNA-seq data, identify the CNV, by use the sc-CNV versus single cell genomics data. I know there's advantages and disadvantages for those two strategies. Could you talk a little bit more about that, based on your understanding?
Right, and we did compare with SC transform. I can't remember whether it made it into the paper or not. The two methods kind of start to diverge pretty early on, and we found our method gave much more consistent results across a variety of different cells in terms of, certainly in terms of the midpoint of gene expression, the transcriptomes, and given that our analysis, our normalization is also based on the high-end expressing genes, I think across the board we were seeing better numbers there. But some of the other normalization methods are very valid as well. It really depends on what sort of analysis you're trying to do. But we just wanted to bring our approach there because it does seem to enhance the midpoint of aligning the transcriptomes across cells, which allows you to really compare gene expression across cells in a slightly more accurate way. And we found that when we went on to do single cell CNV analysis, that the inferred CNV analysis, that having those RNA transcriptomes aligned well gave better results for all cells, whereas with some of the other methods there was populations of cells where the analysis was not working well probably because the cells weren't normalized as well. And then in terms of we would have loved to have done this kind of study using single cell DNA CNV profiling. But at the moment, the technology to do that, as well as do RNA profiling in the same cell, I'm not aware of it being done in a high throughput fashion or sort of being commercially available. I'm sure there are people working on it. So, this is a sort of method that has a number of advantages. You only need to do the single cell RNA sequencing, and then you can infer most of the CNVs pretty readily from that RNA. So it's cheaper and it's faster. At the moment, it gets around the fact that that technology is not widely available. There are approaches that try and do CNV profiling of single cells by DNA sequencing, and then integrate that with single cell RNA profiling of other cells from the same sample. And I think there's probably a lot of merits to those approach. But personally, I think that our approach, where you get the CNV profile from the actual cell that you're looking at, the transcriptome, I think that has allowed us to get very accurate results and really to drill down on what's happening with 1Q. And you can see the precision of the results with many of the gene set enrichment analyses lining up between those two samples that had different genetic backgrounds and were analyzed totally independently.
Right. Great, thanks. I think we have one question from Wanqing. Wanqing, I think you can unmute yourself.
Oh, yeah. Hi. This is Wanqing from Research Computing. Thank you. This is really exciting work. Thank you for presenting. So I'm particularly interested in the CNV detection part. So it seems like this tool works really well at a resolution of 20 to 40 megabase. I was just curious, based on your experience, do you think we can push the resolution to even higher with the current single cell RNA-seq technique?
The limitation is basically the distribution of genes on chromosomes, and so the chromosomes that have more genes, you can get better resolution than that. But then there are other chromosomal regions where the genes aren't as the gene density is lower, and depending on what type of RNA you're sequencing, you may not get as good resolution in those areas. So yeah, that's the short answer. It's hard to push it beyond the resolution of the RNAs that you're actually sequencing.
So based on your experience, so is it just 10x3n sequencing? And if we are looking into gene dense regions, so how high could the resolution go?
I don't have a number answer for you. My guess is it would probably be fourfold better than that in gene dense regions, or and fourfold worse in gene sparse regions at a rough guesstimate. I mean, we've done 3', 5'. The method works for both RNA methods.
Okay, cool. Thank you so much. Yeah.
Sure. Thank you.
Okay. We have one more question from Courtney.
Yeah. Hi. That was an excellent talk. My question is a bit more general about kind of downstream of learning this about these types of cancers in general. So one is, what does it, now that you know what the 1Q duplications are doing, how can that affect choices of therapeutics and down the line, and does this give us better insight into what novel therapeutics might work in these types of cases?
It's a great question, and certainly I'll be discussing the therapeutic implications with some of my clinical colleagues. I do think that it'd be really nice to be able to target oxidative phosphorylation, in these high-risk myeloma tumors. And I didn't show it, but we've done that kind of work in some myeloma cell lines, and in fact, the highest risk myeloma cell lines, one with the most adverse genetics and gain of chromosome 1q, are the most sensitive to the oxidative phosphorylation inhibitors. So, whether there's a therapeutic index for doing that in patients, I think is an open question, and I don't know the answer to that. But it is possible, I guess, if targeting the whole oxidative phosphorylation process is not the answer, then perhaps targeting just one or two of these enzymes that are driving it in the tumors with gain of chromosome 1q, perhaps there might be a therapeutic window to do that.
Yeah, that might be interesting. And kind of related, have you thought, or is it known sort of now that you know the whole pathway, can you imagine how this knowledge might affect sort of more precision medicine? So looking at non-common CNVs that come up, or other types of mutations that might be in the background of some patients. So sorry, I phrased that badly. beyond the 1q duplication, could there be other variants that could be even novel in particular patients that might be worth looking at, like, I guess, duplications of particular respiration genes or something like that?
Sure. We actually found, just as a baseline, that some of the myeloma subtypes had higher levels of oxidative phosphorylation from the get-go, even without 1q. And I personally think that may be contributing to their adverse prognosis as well. So, even if we take 1q out of the equation, there was this other correlation between those subtypes and 414, for example, and poor prognosis and increased oxidative phosphorylation. So it did seem to be like a common denominator for some other lesions that we see in myeloma, which is really interesting.
That's great. Very